Hi to everybody, and I welcome you all to today's lecture. In today's lecture, we are going to study about the pale soft exudative meat and the porcine stress syndrome. In the porcine stress syndrome, I'll be talking about why it is occurring, in which breed it is commonly found, and how to prevent the porcine stress syndrome. And in the case of pale soft exudative, what is the mechanism of the pale soft exudative? What are the disadvantages of pale soft exudatives, and why we cannot use it for the product making? So with this small introduction, let me get into today's lecture. So I once again welcome you all to today's lecture. So porcine stress syndrome is highly stress related conditions. So whenever you put the pig under stress conditions, it can lead to a type of meat called as a pale soft exudative meat. So the incidence of porcine stress syndrome is related to following variables. One is the season. The pigs are prone for stress when the environment temperature is high, say above 23 or 25 degrees centigrade. The pig will start showing the symptoms of high temperature, so it will be put under stress. Even when the temperature is below 10 also, we can find the pigs is coming into a stress conditions. Next is the breed. This is mainly seen, the porcine stress syndrome is commonly seen in three type of breeds, we can say. One is the Poland China, Landrace, and the PIETRAIN are more susceptible to porcine stress syndrome. Again, the sex, the study shows the higher incidence in gilt than in burrows. So in 1960s and 70s, the breeders, to improve the muscling and to reduce the fat, they were doing a selective breeding. So in the young ones or in the next generation, they found that the ox or the pigs which are selected or from the parent through the proper selections, which is having high muscling and less fat, are very prone for porcine stress syndrome, or the meat from such type of animal is having a pale exudative type of meat. Then rough handling, that is when you beat the animal, or when you yell at the animal, when you shout at the animal, when you use the stick and beat the animals, or when you use the electric guards. So all these things will put the animal under fright, which can in turn put the animal under stress. The vaccination, overcrowding of the pigs, all this can lead to pale soft exudative type of meat. And mixing of unfamiliar animals, that is, if you mix the different social groups of pigs, then there can be a stress on the pigs, because pigs always prefer to be in their same social group. When you mix the animals from different pens or different farms in the lairage or during the transportations, that can also lead to the pale soft exudative type of meat. So in the nutshell, I just want to tell you that whenever you put the pigs under acute stress before slaughter, I'll repeat, whenever you put the pigs under acute stress before slaughter, in such type of pigs, we can come across this pale soft exudative type of pork. The breeds in which the porcine stress syndrome is common. One is the Poland China, next is the Landrace, and third is the PIETRAIN breeds. So in this type of breeds, we can come across commonly the pale soft exudative, and this breeds are very much prone for the porcine stress syndrome. So what are the symptoms of porcine stress syndrome? So whenever you put the pigs in acute stress, we can come across these symptoms. One is the open mouth breathing. The tremors of the tail you can see, the tremors of the leg muscles or the back you can see, labored breathing, irregular breathing, then muscle rigidity you can see. It will be inability to walk, respiratory distress, hypothermia. The body temperature will increase. It may increase even up to 41.5, 42 degrees centigrade. So that will result in the cardiac arrest, or generally the breeds which are very much prone for porcine stress syndrome, or the animal which is very much prone for porcine stress syndrome, have a weak cardiovascular system. So the animals will collapse because of the hypothermia, and it can lead to death within 15 to 20 minutes. Similar conditions is also seen in the human. That's called as malignant hypothermia. So it also produces abnormal vocalization. So these are the symptoms of porcine stress syndrome. Let's see a small clipping, the sign of porcine stress syndrome. So you can see here, there is a tremors of the tail you can see, the leg muscles you can see, the tremors of the leg muscles of the back you can see, the inability to walk, respiratory distress you can see, hypothermia, the body temperature will rise to 41.5, the heart failures we can see, and the animals will be collapse. So you can see the tremors, the animal is unable to walk. So it is in the acute stress conditions, and the animal will collapse within 15 to 20 minutes. You can find sometimes the animal will die. So you can see the labored breathing, irregular breathing, you can see in the abdomen, irregular breathing you can see. So why in some breeds the porcine stress is very common? It is because of the molecular defects. So molecular defects means due to the mutation of the ryanodine receptors. We have seen that in the muscle contraction classes, where the ryanodine receptors is the one which regulates the calcium levels in the cytoplasm or the sarcoplasm, because it regulates the release of the calcium from the sarcoplasmic reticulum into the cytosol. Okay. So if there is any mutation occurs in the ryanodine receptors, generally due to the mutation of the ryanodine receptors at arginine at positions 615 on the molecules we can see. So it will not able to regulate the calcium. So calcium will leaks out more rapidly. So when the calcium leaks out more rapidly, what happens is the muscle contractions will be taking place at very rapidly without nervous signals. So the elevated systolic calcium causes the activation of the myofibrillar ATPase. So when the myofibrillar ATPase becomes more active, it leads to accelerated glycolysis. So the pH of the muscles will fall. There will be hypothermia will be there because of the rapid muscle contractions also. So this will put the animal under stress, and ultimately, if you are not taking care, it may lead to death of the animals. So this is what happening in the porcine stress syndrome. So this so I'll repeat once again, it is because of the molecular defects, there is a mutations at the ryanodine receptors, so calcium leaks out more rapidly, and this result in the continued contractions without nervous signals. So a very rapid glycolysis will be taking place in an attempt to replenish the ATP that is being broken down. So in the human, a similar condition is called as a malignant hypothermia. So people often with this condition die or becomes dangerously overeaten when they are given certain anesthetic like halothane. Okay, so this is called as, or this is how the porcine stress syndrome is taking place. So let's see what is happening in the porcine stress syndrome. So when you put the animal under intense stress or acute stress, due to inherent defects in the mechanism for the uptake, storage and release of calcium in the muscles because of the mutation in the ryanodine receptors, it will result in the increased muscle activities and the muscle metabolism changes from aerobic to anaerobic glycolysis. Because of the anaerobic glycolysis, the muscle pH changes to acidity and the body temperature rises to 41 to 41.5 or 41.6, or even it goes up to 42 degrees centigrade. So that leads to malignant hyperthermia. So we can see the different signs like tremors of the tail, the back, leg muscles, rapid breathing, hypothermia. So finally it leads to the collapse and the death can occur within 15 to 20 minutes, and the rigor mortis occurs rapidly within 5 to 10 minutes. So this is what happens in the porcine stress syndrome. So why the PSC occurs? So any acute, that's what in the last slide we have seen, any acute, it may be intensive and very short term pre-slaughter stress like panic, mixing of social groups, rough handling, etc., which we have seen in the previous slides, result in the flight or the flight reactions. So because of this flight or the fight reactions, increase in the metabolisms. So increase in the metabolism, body metabolisms to maintain the homeostasis will be taking place. So increase in the body temperature will be taking place. So utilization of the body glycogen and its fast breakdowns. Due to the fast breaking down, there is a high accumulation or the fast accumulation of the lactic acid take place and the pH will drop rapidly. So there is a quick decrease of pH after the slaughter, but at the same time the carcass temperature is high because of the hypothermic conditions. So this is the PSC meat. We can see it is very pale, soft, and it is exudative. This is the normal pork, and this is the pale, soft, exudative pork. You can see the color difference. Okay, so this is very pale, it will be very soft, and it will be very watery. So you can find the water will oozes out. So let's see how the PSC or the pale, soft, exudative meat is formed, or how does the porcine stress syndrome affect the meat quality, or how does the acute stress before slaughter affect the meat quality. When you put the animal under acute stress, the fight or the flight reactions will be taking place. To maintain the homeostasis, the glycolysis will be taking place very rapidly, which you have seen in the previous slides. So there is a sudden drops in the pH, that is within 45 minutes to 1 hour, the pH will drop suddenly. So normally, if we take in the pigs, to attain the ultimate pH of 5.6, it will take around 8 to 12 hours. So as the pH decreases, the temperature will also decreases slowly. So when it attains the pH of around 5.6, we have seen in our meat technology unit, the temperature of the carcass is around 10 to 15 degrees centigrade. But in the case of PSC meat, what happens is there is a sudden drop in the pH. Within 45 minutes to 1 hour, there will be a sudden drop in the pH. So the ultimate pH is attained within 45 minutes to 1 hour. But the carcass temperature is high. I'll repeat it. The sudden drop in the pH within 45 minutes to 1 hour, but the carcass temperature is high. Because of the high carcass temperature and sudden drop in the pH, there is a more denaturation of the protein than the normal pork. So denaturation of the protein is very high. The length of the myofilament is reduced to around 8 to 10% during this process of denaturations. There will be precipitations of protein, especially the sarcoplasmic proteins will be taking place. Because of the denaturation of the proteins, the water holding capacity will be lost. So in the DFT conditions, we have seen how the water is held in the muscle fibers. Because of the denaturation of the proteins, this muscle proteins will lose the water holding capacity. So the meat will start losing the fluids or large quantity of fluid will come out. So it is a exudative conditions. And lot of nutrients will also, water soluble nutrients will be also lost along with this exudates. So the muscle will look pale, soft and exudative. So 95% of the porcine stress syndrome pigs will produce the pale, soft, exudative. So let's see how to avoid the porcine stress syndrome. So one is the gentle handling of the animals. You spray the water. So when the animal, you find the body temperature, when the environmental temperature is increasing, you spray the water. Cool water can be sprayed. Proper ventilation should be provided. So loading and unloading, during the loading and unloading, take care. Use proper ramps. Proper slope of the ramp should be there. The lightage design should be proper so that it helps in the easy movement of the animals. So the animal will not be put under stress. Maintain the social groups. Maintain the social groups. And providing water and food in the lightage. So you have to provide sufficient quantity of water. Avoid using of avoid using of the stick. Use proper electric, do not use the electric goats. Separate the aggressive animals. So in the meat technology also we have a separate, when we find any animal is very aggressive, we separate that animals from that group. Then you can feed the sugar or the jaggery before transportation. So you feed the sugar or jaggery, that is molasses, you can feed in liquid form to the pigs at least 3 to 4 hours before transportations. This also will reduce the PSC conditions, and it also helps to reduce the shrinkage loss. So withheld feed for 4 to 6 hours before transportations. Careful selection of the stock, that is what I told you. When you are doing the breedings, you have to do carefully. When we concentrate more on muscling, it can lead to the porcine stress syndromes also. So how to avoid the porcine stress syndrome? So porcine stress syndrome is caused by the recessive gene when it is in the homozygous condition or the heterozygous conditions. So when it is in the homozygous or heterozygous condition, the recessive gene can leads to this porcine stress syndrome. So how to do that? How to select the particular animal for breeding? So in the age of 7 weeks itself, we can select it. We can use a test called as allothane gene test. So you know that allothane is a inhalant anesthetics. So when you allow the pigs to inhale, so we find if the limb is became very rigid, then we can say that it is very much prone for the stress conditions. Another is if they are not recovering from the anesthetic condition within 45 minutes to 1 hour, again such types of pigs is also very much prone for the porcine stress syndrome. And now the latest technology is DNA based technology we can use to detect before going for breeding of the selecting the parent stock for the breeding. So if you come across such type of pig porcine stress syndrome, we can also use this dantrolene sodium. IV you can inject it, so that is act as a muscle relaxant, and we can help to prevent the death of the pigs due to porcine stress syndrome. This just for your knowledge, I'm just put here. So dantrolene sodium IV you have to give. So this is help to overcome it help to prevent the death of the pigs due to porcine stress syndrome to some extent. What is pale soft exudative pork? So this is, you can see it is, the color is very pale, it is not attractive. The meat will be very soft and it will be exudate a lot of fluids. So you can see a lot of fluids will start coming out of the meat. The pale soft exudative meat is found in carcass from animals which just before slaughter have undergone intensive or acute stress conditions. This meat is quite pale, soft and watery. and there is a little evidence of marbling. The marbling you cannot see in, or there is very little evidence of marbling, and it is mainly seen in pork. So let's see what's the difference in the glycogen content. So in the normal animals, already we have seen in the DFT, during the DFT class discussions, we have seen it is 1%. In the DFT, it is 0.3%. In the pale meat, in the PSC, the glycogen content will be around 0.6 just before slaughter. Glycogen at 24 hours, it is 0.1, 0.1, 0.1%. Lactate productions, high, low. It is also mostly, it will be normal with that of the, it will be equal to that of the normal pigs. But the problem here is there is a sudden fall in the pH when the carcass temperature is high. So once again, I will explain it to understand better why the PSC is occurring. So whenever we put the pig under acute stress, there is a sudden drop in the pH, that is within 45 minutes to 1 hour, the pH will drop suddenly, but at the same time, the carcass temperature is above 35 degrees centigrade. The combination of these two, that is sudden fall in the pH plus high temperature of the carcass, will result in the denaturation of the protein and precipitation of the soluble proteins like myoglobin. So denaturation of the protein is more than in the case of normal pigs, though the pH is attained in both are almost same, but here in the normal pigs, as the pH decreases, the body temperature is also decreases. So the denaturation in the case of normal pig is very, or comparatively very less than compared to that of the PSC meat. Because of the denaturation of the protein structures will be lost. It will not be firm. It will be very loose-like. It will be very soft. And because of the denaturation of the protein, it will not able to hold the water. The water will start coming out of the muscles. So it will be very exudative. Because of the denaturation of the protein structures will be lost. The protein fibers are held very loose. So there is lot of intercellular spaces, or interfiber space will be more. So more surface to reflect. So whatever the light that falls, it will be reflected. So it will be soft, it will be pale, soft and exudative. So I hope it is very clear. So why the consumer reject the pale, soft, exudative meat? One is, it is pale, so naturally it is not attractive. It will lack the flavors because most of the water-soluble nutrients will be lost, and whatever the curing ingredients we add, it will not get absorbed, which I will be explaining in the coming slides. It will have a very soft, musty texture. The texture will be lost because of the denaturation of the protein. The watery and exudative, the meat lacks juiciness and it is dry on cooking. So juiciness will be lost because all the moisture will be lost because of the denaturation of the protein, and it is not suitable for curing. So how to identify the PSC by the consumer? One is the color. So it will be pale, soft and exudative. So undesirable appearance and shrinks excessively. So this is how we can identify is one by the color. So another thing is, you can, as a technologist, what we can do is we can see the color, as I told you here. We can see the color and you can compare it. You can compare this color. You can see this pale, grayish pink like. Okay. This will be the pale, soft, exudative meat. So you can compare with the color standards. Another is, you can check for the pH. So within 45 minutes to 1 hour, if you check the pH will be around 5.6 or 5.8 like. the pH will fall very rapidly. But in the normal pH, if you want to attain the pH of around 5.6, it takes around 8 to 12 hours. So here we can check within 45 minutes to 1 hour, use the prop pH meter and just check the pH of the meat. You will find it has reduced to 5.6 or 5.8 like within 45 minutes to 1 hour. So rapid fall in the pH. So you can see here in the ribeye area, this is a normal pork and this is the PSC pork. So you can see between the 10th and the 11th ribs. Same things we have seen in the DFT meat between the 12th and the 13th ribs. In the case of pigs, it is between the 10th and the 11th ribs. So you look for the color, firmness and wateriness. If it is watery, then it is exudative. If it is having a very pale color, which you can also compare with the color standards, and when you touch it out, it will not have the firm structure. The firmness will be lost. So this is how we can evaluate whether the meat is PSC meat or not. Why the PSC meat is pale? Already I told you, because of the sudden fall in the pH, when the carcass temperature is high, there is a denaturation of the protein. So the muscle fiber proteins will shrink because of the loss of water also, resulting in the large gaps between the individual fibers. So there is a large gaps in the individual fiber, just like exactly what is happening in the DFT, opposite of DFT. Okay, here the resulting in the large gaps, we know that in the DFT meat, it is swollen and the muscle fibers are very intact, there is no space. But here we find there is a large gap between the individual fibers. And small myofibrillar volumes causes open meat structures. So there is more open space. So muscle tissues with smaller myofibrillar has high light scattering ability. So whatever the light, because of the more space or the surface area, the light scattering will be more. The light is unable to penetrate into the meat. So the muscle becomes scattered right on the surface. The myoglobin cannot absorb the light, thus making the meat appear pale. You know that the light is unable to penetrate into the meat, so becomes scattered right on the surface. So when the light falls, immediately it will be scattered because it has more surface area, because of the large gap between the individual fibers, and the myoglobin cannot absorb the light, thus making the meat appear pale. Why the PSC meat is soft and less firm? So again, it is because of the denaturation of the protein. Denatured protein exhibit a change in the three-dimensional structures. So native protein will be lost. So it loses the three-dimensional structures, which leads to less firm structure overall. So overall, we find it will become very soft. Okay, it will become very soft. It is not again desirable for the consumers. Why the PSC meat is watery? Sudden fall in the pH with high carcass temperature. So denaturation of the protein. So more denaturation of the protein. So smaller quantity of native proteins. So it will not able to hold the water. So it is highly exudative. So you can see here, this is the normal, this is a PSC pork. You can see the exudative, the fluid that coming out is very high. This is the normal pig's pork. You can see here the fluid is very less. So this is very exudative or watery. So all the water in the muscles will be coming out. You know that 70 to 73% is the water. So most of the water will come out. So that is why we call it as a exudative. Why PSC meat is a concern for curing industries? So as I already told in the DFT, the water has to come out and absorb whatever the curing ingredients you add. Let it be the salt, nitrates, or if you're adding the spices, whatever you had, it will come, the moisture from the meat will come out, it will absorb and it will penetrate inside. This is what happening in the normal meat. Here what happens is, since it has lost its water holding capacity, the water will just comes out, it will not get absorbed back. So whatever we add, it will not get absorbed. That is the issues with the PSC meat. So that is why we cannot convert this meat to curry or other things, because we can change the color when you make the curry, but problem is that the meat will not, the curry will not taste. So it cannot absorb the curing ingredients. To some degree, I told the myoglobin is also denatured in the PSC meat. The precipitations of the myoglobins will also take place because of the rapid fall in the pH and the carcass temperature is high. The precipitations of the water-soluble proteins will also take place, like myoglobin will be taking place. So since the denaturation of the myoglobin, it will not able to form the cured meat color, because you know that nitrosohemochromogen, for the formation we also require the myoglobin. So the myoglobin will also be denatured, so it will not be suitable for curing. And in the emulsion products also we cannot use it because it has very less emulsifying capacity. So you cannot use for the sausages and other things also. So this is the disadvantages of the PSC meat. The PSC is also known as watery pork, is when the ultimate postpartum measured after 45 minutes is less than 6. So here you can see is that we have already seen in the DFT classes, the meat with pH at 45 minutes being lower than 6. So within 45 minutes to 1 hour, the pH will be around 5.8 to 6 or 5.6 like. Okay, so it's a rapid fall in the pH. So just check the pH of the meat, and if the pH of the meat within 45 minutes to 1 hour is less than 6 means it clearly indicates it is a PSC meat. The meat with ultimate pH of 5.3 or 5.4 like or 5.5, it is just like a normal pH only, but the problem here it is falls very rapidly. So it leads to more denaturation of the protein than in the normal meat. What is red soft exudative in pork? The red soft exudative, it is also called as a NAPOL gene effect on pork quality. So this is due to the homozygous or heterozygous NAPOL gene that is present, which will be leading to these conditions, that is red soft exudative. Here the pork is red as normal pork, which is generally preferred. The red color, it will be more red color, which is preferred by the consumer. The PSC pork, so red soft exudative pork, has a red color that consumer desires, but it is soft and exudative, indicating that the muscle protein have low water holding capacity. So you can see here it has a red color that consumer desires, but it is soft and exudative, indicating the muscle protein have low water holding capacity. So this is the PSC pork. This is the red soft exudative pork, where the color is red like a normal pork, but it is soft and exudative. So why it is occurring? It is mainly seen in a particular breed that is called as a Hampshire breed. This is the Hampshire breed. Why it is occurring? See here, the NAPOL gene, that is in the homozygous or in the heterozygous, will have a modified adenosine monophosphate kinase. So you know that adenosine monophosphate kinase is the one which regulates the glycogen synthesis. If this is not active, or if there is any alternation in this adenosine monophosphate kinase, it will not able to regulate the glycogen synthesis. So in the case of red soft exudative, what happens is, because of the homozygous or heterozygous conditions, there is a modification in the adenosine monophosphate kinase. So it will not able to regulate the glycogen synthesis, and it will not able to effectively inhibit the glycogen production as in the normal animals. So the glycogen accumulation will be more. Since the glycogen accumulation is more, the pH drop will be high. Here the pH will also drop slowly only, but the quantity of lactic acid formed will be more. Because of the low pH, it will come around 5 or 4.5, like very low pH than the normal. It will lead to denaturation of the protein. because of the denaturation of the proteins, it will not able to hold the water, and because of the denaturation of the protein, as I explained in the PSC conditions, it will lose the firmness, or rather this meat will become soft. So this is what happening in the red soft exudative conditions. So what are the disadvantages? Lower processing yield, because it is very exudative, the fluids will come out. It has a lower processing yield. Higher drip loss in cooked cured meat. Again, it has very lower water holding capacity. The fluid loss will be more. Low marbling score, similar to that of in the PSC pale soft exudative. It will also have the low marbling scores. What are the advantages of this NAPOL gene? Higher daily gains, because the glycogen accumulations is more, so it will give higher daily gain. Higher lean meat content, the meat content will be more. Larger proportion of ham, the ham is larger. Add less side fat, the side fat or the subcutaneous fat will be less. So if you see the combination, no variation in the combination of carcass is found. Just like in the normal pork, the combinations, the composition is same to that of the normal pork. The rate of pH decline is also normal. Here, just like in the normal pigs, as the temperature decreases, the pH will also decreases. But the problem is that the pH is much lower than in the normal pigs. In the normal pigs, the ultimate pH will be around 5.4 to 5.6. Here it can come up to even up to 4.5 because of the more glycogen content. So this is the red soft exudative in pork. So let's see the difference between the PSC and DFT meat. As I already described, so we are in the nutshell what we studied in the PSC and DFT, we are going to see here. It is because of the short duration stress before slaughter. It's a long duration of stress. Because of the short duration stress before slaughter, because of the fright or the flight mechanisms, it will accelerate the postmortem glycolysis. So result in the rapid fall in the pH when the carcass temperature is high. So that will lead to denaturation of the protein. Here, when you put the animal under long duration of stress, the glycogen resource will be very less. So it will not attain the ultimate pH of 5.6, rather it will be around ultimate pH will be more than 6. So here it is pale, soft and exudative because of the denaturation of the protein. Here there is no denaturation of the protein because the pH is high. So small extracellular space, it will have more extracellular space. So more will be the reflection of light. Here absorption light will be more because there is less surface to reflect. Protein denaturation is high, the protein denaturation is less. Larger extracellular space, here smaller extracellular space. Here the spoilage will be faster. Here the spoilage will be normal or low. Low water holding capacity, here it will have a high water holding capacity. It scatters more light back to the observer. It scatters less light, so it is dark in color. It scatters, since it scatters more light, it is pale in color. Reduction in absorption of green light by the myoglobin. The oxygen used by, most of the oxygen is used by the cytochrome enzymes, so less oxygen is available for the myoglobin to form oxymyoglobin. Here the denaturation of the myoglobin will also take place. So the oxygen, the cured color cannot be formed. The meat looks less red. The meat looks also less red. So thank you very much for listening to this class. Take care. Be safe. God bless you all. Thank you.